Good morning, everyone. In this video, we will learn about spread plate and pour plate methods. These are two important methods to isolate the bacterial colonies and also helps to count the bacterial colonies which are growing on the bacterial growth media. So, one is pour plate and another is spread plate. These are two methods, and the third one is stick plate, which I have already discussed about in my previous video. Now, in case of pour plate method, we have to collect, suppose we want to isolate the bacterial samples from the soil. So we have to collect the soil sample first and make several dilution to reduce the microbial count in the cell in the sample. Okay, suppose we prepare the stock, that is 1 gram soil in 10 ml saline water. This is a stock. If we plate this one, it will give rise to different types of bacteria, but that are in a huge in numbers, so we can't calculate them or we can't count them. So if we want to get a proper number of the colonies present in this soil sample, we have to dilute the sample, like 10 to the power minus 1 dilution, 10 to the power minus 2 dilution, 10 to the power minus 3 dilution, and so on. Then only we can get a proper count of the bacterial colonies. Okay, so in pour plate technique, after preparing the dilutions, what we will do, normally inoculum from the dilution is mixed with the soft agar, okay, which is in the semi-liquid form. Then this is poured in the Petri plate containing the growth media. Now, after incubation at 24 degrees centigrade and 24 hours, and 37 degrees centigrade, it will give rise different colonies. Okay, we will see different colonies in the agar plate, but in this case, it is very important for pour plate method. The colonies are usually embedded, or it actually helps to embeds the bacterial colonies within the agar media. Okay, there is agar media. It's a agar media like this. It's a soft agar. So we first, from like 10 to the minus 2 dilution, 100 microliter or 0.1 ml inoculum must be added into this. Mix it well and then pour it and mix the plate with clockwise and anti-clockwise direction and let it for incubation. And after 24 hours incubation, or sometimes for 48 hours or 72 hours, depending on the type of bacterial colonies which are growing on this plate, the bacterial colonies will be grown on this media, but they are embedded in the agar. Okay, they are embedded in the agar. So this is the basis of the pour plate technique, but with the help of this technique we can count these colonies. Okay, now the spread plate technique. This is another one. Spread plate technique basically use the same procedure from the soil sample collection, then prepare the dilutions, etc. Finally, after the dilution, suppose from 10 to the power minus 1, 10 to the power minus 2, 10 to the power minus 3, these type of dilutions have been made from the stock soil sample solution. If we want to plate from 10 to the power minus 3 dilution, what we will do? We just take 100 microliter or 0.1 ml inoculum and spread it on a bacterial agar media. How? Like this. We just spread it onto the bacterial agar plate and then incubate for 24 hours and 37 degrees centigrade. This is a general growth condition for bacteria. Now, for this spreading, we need a spreader, a glass spreader usually used. Otherwise, heat resistant different types of plastic spreaders can also be used. Now, how the spreader looks like? The spreader looks like this. Okay, this is a spreader. This basically, it is not straight. The spreader basically have this type of shape. So with the help of this spreader, we have to hold it like this and spread the bacterial samples on the plate. First, we have to add the inoculum onto this and then spread it with the help of the spreader and keep it for the incubation. And after incubation, what we will get? We get different types of colonies growing on the solid surface of the media. In case of pour plate technique, the colonies are embedded within the media, and in case of spread plate technique, the colonies are appeared on the surface of the bacterial growth media. So this is a very minute differences which we have to remember. But both this bacterial plating method helps us to count the different types of bacterial colonies which are present in the dilution, and after multiplying it with the dilution, we will get the actual number of colonies from the original soil sample or soil sample or water sample, whatever. So these two plating techniques are also useful as the streaking plate technique, though streak plate method is generally used to get a pure isolated, well isolated colonies. So these three types, spread plate, pour plate and streak plate method, are very, very important in microbiology to get an idea about the number of colonies present in the natural source, and also to get a well isolated colonies with the help of streak plate method. Hope you like this video. Thank you so much.